Welcome to solverfy.
Hello students, for the given question, first part, the process used by bacteria to break down and utilize lactose molecules is called lactose utilization. To break down lactose, Escherichia coli would first need to transport the disaccharide across the cell membrane. This is accomplished by the action of the enzyme lactose permease, which is encoded by the LAC-Y gene in the lac operon. Once inside the cell, lactose is cleaved into its constituent monosaccharide glucose plus galactose by the enzyme beta galactosidase, which is encoded by the LAC-Z gene. The glucose and galactose can then be further processed through glycolysis to produce ATP. For the second part, Listeria monocytogenes is a glucose metabolizing facultative anaerobe, meaning that it can use both aerobic and anaerobic pathway to produce ATP. If oxygen is present, aerobic respiration would create the most usable ATP for the bacterium. This process occurs in mitochondria of the bacterial cell and involves the following steps: glycolysis, the citric acid cycle, and oxidative phosphorylation. The final products of this process are ATP, water, and carbon dioxide. If Listeria monocytogenes does not have access to glucose and must metabolize triglycerides or protein instead, the steps of the pathway would change. For example, triglycerides would need to be broken down into glycerol and fatty acids, which can then be processed through beta oxidation and the citric acid cycle to produce ATP. The third part, lactic acid fermentation, is the process by which leuconostoc mesenteroids and other anaerobic bacteria convert glucose into lactic acid. This process occurs in the cytoplasm of the bacterial cell and involves the following step: glycolysis, where glucose is converted into pyruvate, and reduction, where pyruvate is converted into lactic acid by the enzyme lactate dehydrogenase. This process is important in the fermentation of foods such as pickles, as it helps to preserve the food and create the tangy flavor associated with these products. For the next part, fourth question, alcohol fermentation is the process by which Saccharomyces cerevisiae and other anaerobic yeasts convert glucose into ethanol and carbon dioxide. This process occurs in the cytoplasm of the yeast cell and involves the following steps: glycolysis, where glucose is converted into pyruvate; oxidative and decarboxylation, where pyruvate is converted into acetaldehyde by the enzyme pyruvate decarboxylase; and reduction, where acetaldehyde is converted into ethanol by the enzyme alcohol dehydrogenase. This process is important in beer production as it is responsible for the alcohol content and carbonation of the beverage. I hope you find the video useful. Thank you.